nervous system stem cell. Our proprietary name is HUCNSSC, human central nervous system stem cell. This cell is restricted to the CNS lineage. It is hardwired, if you will, to differentiate only into the progenitor cell types, astrocytes, oligodendrocytes, and neurons that go to make up the central nervous system. These cells expand in culture, and so are scalable on demand. They stay in the stem cell compartment in a cryopreserved state, and then differentiate or transplanted in vivo into the host. This model is an allogeneic approach. It's not autologous. The cells are not genetically modified, and there is no in vitro pre-differentiation required prior to transplant. Some of the important attributes of the cells that guide us in terms of our thinking on translation. Number one is that the biological activity of the cell is regulated by the host. The cells self-renew, and they are non-tumorigenic. They survive in vivo, differentiate, and migrate again in vivo. And the mechanism or putative mechanism of action would be described as either cell replacement or cell protection. So in the animal world, we made a number of important observations that, again, helped inform us and guide us in terms of our selection and choice of disease targets. So in a lysosomal storage disease model, we found that these cells were neuroprotective, and I'll talk some more about that in a moment. We also saw that these same cells myelinate hypomyelinated axons in what is probably one of the most famous mouse models on the planet, the sugar mouse, certainly for those who are looking at myelination disorders. Moreover, this cell restores lost motor function in spinal cord injury and preserves vision in models of retinal degeneration, specifically in a model of retinal degeneration known as the Royal College of Surgeons rat. And last but not least, we've recently observed with our collaborators that these cells restore lost memory in the triple transgenic and the CAM-TET DTA mouse. So our first at bat was a lysosomal storage disorder, neuronal ceroid lipofuscinosis, which is a mouthful. We were focusing on two forms of the infantile version of NCL. And this was based on work that was conducted by the scientists at the company, in which they were able to demonstrate that they could cross-correct for a genetic deficiency, whereby in the animal, the host neurons are enabled to lyse or get rid of toxic cellular waste that kills off the neurons. So by transplanting the healthy cells, they were able to cross-correct because the diseased host neurons were able to take up the missing enzyme. In this case, two different variants, the PPT1 and the TPP1. And the mechanism is described as uptake through the mannose-6 phosphate receptor. And this work was featured in Cell Stem Cells, some very, very excellent work by the scientists at the company. So we did the clinical study in NCL, commonly referred to as BATTNS. We executed a phase 1 study, and we learned a lot from this first study. We learned that, and we did so because this was a very aggressive disease, and we were transplanting into the most severely impacted patients. So 3 of the patients succumbed to the natural progress of the disease, and we were able to get our hands on the tissue and do autopsy. And so, as a result, not only were we able to see that the cell survived for over 2.5 years post-transplant, but an important question was what happens when you stop immunosuppression. And we observed in at least one of these patients that 1.5 years post-withdrawal of immunosuppression, the cells were still viable and evident. What was interesting too was where we found the cells, and you can see these red asterisks. This is where we found the cells, and here's where we transplanted them in this hemisphere. And you can see that the cells migrated deep into the brain structure, and that was an important observation for us. And last but not least, in this clinical trial, we transplanted over almost a billion cells into the brain in a very, very sick patient population. We came out of that study with a very good, very favorable safety profile. We filed a protocol to do a second study in the less severely impacted population. But after about 6 months of trying to find those kinds of patients, those who were less severely impacted, who had most of their neurons intact, we essentially decided to shelve that study because it was taking too long for our healthcare system to actually do a proper diagnosis on these children. And by the time they were diagnosed, maybe 2 years plus post-birth, most of their neurons had been damaged. The second at-bat was in a myelination disorder, and this was based on work that was done with, This was based on work that was done in collaboration with the folks at OHSU, Stephen Back, and our scientists in the company. And what they showed was that when you transplant these cells into the sugar mouse, that you get new functional myelin. This is published now in Science Translation Medicine. You see differentiation of the cells in autologous dendrocytes, and we saw myelin-enhanced nerve conduction in these animals through very objective measurements. And we used MRI to detect human myelination in the animal model. It's much easier to detect human components and proteins in an animal than it is to detect human cells in the human. So the idea of being able to use myelin to use imaging to evaluate the potential for generation of de novo myelin is obviously very appealing in terms of the clinical trial design. So we went on and we did a study at UCSF, phase 1 study, where we transplanted 4 children with these same neural stem cells. And using MRI, we're able to show that there was evidence of progressive and durable myelination. And there were also small but measurable gains in neurological function. This is somewhat unexpected given the natural history of this disease, and it's a very aggressive disease, and these children go downhill very quickly. We also saw other signs of clinical effect that's helping us in considering the design of a phase 2 study. So the results of both of these efforts, the preclinical and clinical, appeared back-to-back recently in Science Translation Medicine. The FDA approved study involved 4 patients with the early severe clonatal form of the disease. The children were aged 6 months to 5 years of age. We transplanted bilaterally into the white frontal lobe. We put in a total of 300 million cells, and we immunosuppressed the children for 9 months. So the endpoint's obviously #1 is safety. We were also looking for evidence of some kind of a biological effect. And we were looking for detection of de novo myelin using MR diffusion-weighted imaging. So the results I've just mentioned are published in October 2012 in Science Translation Medicine, and a phase 2 study is now in the planning stage. So this is a very important observation for us in the company, that we knew from the first study that the cells engraft and they do survive, they do migrate, and they do maneuver. But we didn't know if they were biologically active. We now got an answer to that as a result of this study, where we see the generation of myelin and the enduring effect of the cells over time. The next study that we engaged in is in thoracic spinal cord injury. This is a study that's going on currently in Switzerland at Bauer's Hospital in Zurich. And all of the excellent preclinical work that supported the decision to go into a clinical trial in spinal cord injury was conducted in collaboration with our great friends and collaborators at UC Irvine, Aileen Anderson and Brian Cummings. And essentially what they showed was that these human neural stem cells differentiate and promote locomotor recovery in spinal cord injured mice. Interestingly, they also showed that those animals who had regained function, when you ablate the cells, take away the human cells, they lose that restored function. So evidence perhaps that the cells are directly implicated not only in the recovery, but also in the maintenance of that function. The phase 1/2 study in Switzerland open to patients in Europe, Canada, and the US, enrolling 12 patients with thoracic injury, T2 to T11. This is not the acute phase of injury. It's probably more towards chronic than acute. And we only seek to enroll patients who have at least 3 months between them at the time of the injury, and not longer than 12 months post-injury. And we do so because, obviously, one of the things that we're interested in seeing is the effect of the cells, and anything that would create noise by the natural progression from injury of what's called an ASIA A, of the most severe to a B, might confound the results of the study. So we go in after 3 months. The design of the study is progressive in that in the same study we have an opportunity to start off with the most severely impacted and work our way through to less severely impacted, the Bs and the Cs. We are putting a significant cell dose into the spinal cord, and the cells are deposited above and below the lesion. Again, safety is job one for us here, but we also have endpoints where we're looking for clinical effect, and we're using electrophys and quality of life endpoints. The ASIA A cohort dosing was completed in December last year. We presented the interim data at the International Society for Spinal Cord Injury in the UK in September of this year, and the first ASIA B patient was dosed in October. And what did we see? Well, we saw that continued good safety profile, no toxicity associated with the cells or the immunosuppression regimen or the surgery. There were no adverse events attributable to the cells. And interestingly enough, we observed in this worst of the worst patient population, there were changes reported in light touch sensation. This can be very, very subjective. However, the quantitative perception testing and electrophys and other measurements show significant segmental gains in these patients. So that is an unexpected observation, not because we don't have faith in the platform, but because you do not expect a gain in these patients at that stage of injury. The next field of endeavor is in the eye, in age-related macular degeneration. I'm not going to bore you with the statistics, but it is the leading cause of vision loss in folks over the age of 55. The preclinical work was done with Ray Lund at OHSU. His work was featured in the European Journal of Neuroscience, and very, very rigorous preclinical research. But essentially, it led us to believe that perhaps these cells could be useful in AMD and might be neuroprotective. So we chose to go into geographic atrophy or age-related macular degeneration. Open label, dose escalation study, 2 cohorts. The first cohort of 8 subjects essentially are legally blind. We're just dosing into the worst eye, and so the nice feature about doing this is you have a degree of control using the untreated eye. Again, the objectives here for job 1 is safety, but we're looking for preliminary efficacy. And because this is an external organ, we have an opportunity to do some very nice measurements and performance evaluation of the treated eye versus the untreated eye. And the data can be obtained relatively quickly. So, just to sum up our clinical progress to date, we have 2 trials in patients completed for brain disorders, and a 3rd is underway in spinal cord injury. Yes, these are small, uncontrolled studies, but there are signs of clinical effect emerging that are in fact confirming the observations in our preclinical studies. We're now moving to much larger targets, such as AMD and Alzheimer's disease, and taking on bigger challenges and bigger risk. The cell dose ranges from 200,000 cells in the eye to over 1 billion cells in the balanced brain. So the prospect here is for an enduring clinical benefit from a one-time intervention with the cells, because they do integrate into the host, either alone or in combination with other treatment regimens. These are the classical targets that one sees for cell therapy. I'm not going to go through them all. But essentially those that you see in blue are programs where we either have clinical activity underway or preclinical activity underway. These areas are of interest to us for the translation endeavor. It's never too early to start talking about your label, start thinking about what you're shooting for in time. So one putative label that we came up with was as follows: a neuroactive, cell-based therapeutic for restoration and preservation of neurological function in a broad range of CNS disorders. And as we've seen in the endeavors in LSD, which is neuroprotective, myelination disorders, which is neuroprotective, spinal cord injury is function restoration, retinal degeneration, which is neuroprotective, and then last but not least, Alzheimer's, the goal of which is to restore lost memory function. So thank you very much for your time, and happy to answer any questions you have, either here or outside over a cup of coffee. Thank you.